So who are we? Well, there's just so you get an idea of the size of our crop science unit. There's a professor, a docent. We're going to be recruiting a lector. We have a researcher and another person being recruited, 2 postdocs, 2 PhD students, 2 visiting PhD students, and other masters and other students. Just so you get an idea of how big we are. And of course, like everywhere, we go up and down depending on what's happening and so on. And for those who haven't been to Umeå, we have a campus right in the city. It's situated with Umeå University, which is quite a big university. I don't know how many current students, but maybe 30,000 students, although they're not all on campus at the same time. And we just have a small campus where the forest faculty is the biggest faculty. And one of the really good things is the field station is really close. So it makes it really nice for being at the field station a lot during the summer. And so it's very convenient. And one of the things you'll notice about this picture is that the city's sort of being built around the field station. A number of years ago, some of the land was sort of taken to build an IKEA. And at the moment, they're building a ring road, which is taking off a little bit of the farm. So we did have more than 300 hectares, and now we have a little bit less than 300 hectares. But we still have a really good research farm in a peri-urban setting. And we also, in addition to small research plots, we have big fields which are managed in a conventional way to produce barley and particularly produce forage to feed the animals in the dairy research farm. So, I mean, other people could correct me on the exact numbers, but maybe 120 breeding cows. And we have long-term experiments. Tammsalen is a sites station, so Swedish infrastructure for ecosystem sciences. It's one of the 2 SLU agricultural sites station. So that's really a good thing for our department in terms of the resources it gives us and also the data sets that we can generate. So that's sort of the setting of where we are. And a lot of our research focuses on this, which is forage. And the reason for that is that 70 to 80% of agricultural land use in northern Sweden is forage. It's our most important crop to produce feed for the dairy industry, but also for other ruminant industries. And actually, there are a lot of small beef farms in northern Sweden and some sheep farms as well. So forage is important, and a lot of our research is forage focused. This is a bit of an outline of what we work on, and because our the title of our department is actually says a geographical location, agricultural research for northern Sweden. We do focus on that, and so it's not just one area of crop science. We're quite diverse in the things that we do. And so we've tried to focus on more traditional agronomic research, the use of agroecosystems models and sensor technologies for the purposes of decision making in relation to farmers, and also for developing sustainable agricultural systems. And a lot of our past and present research projects fall on this spectrum somewhere, and often they're sort of in the middle between different areas of research. And so what I'm mostly going to be doing is giving some examples of the type of research that we're doing so that you get an idea, and hopefully that there will be ideas for collaboration, because that's probably the main reason that I'm here. I do already collaborate with people from the VPE department, but I think there's always room for more collaborations. So in terms of agronomy, the variety trials are something that are really important, but they're not actually run by the crop science group. So we have the field station group, and they manage all the variety trials, but they're important because they're a good resource for us to do research. We can sort of piggyback on this variety trial infrastructure that happens every year in barley, oats, grass, and clover. So they're important for us, and they happen. The official variety trials in northern Sweden happen at 4 sites. We manage one at Tammsalen, and then we coordinate what happens on the other research stations. This is this is not a northern Sweden project, but we've recently finished a project on lucerne establishment. And the idea behind this was to consider some of the places in Sweden that we could be growing lucerne, but we don't because establishment often fails. And in the picture on the left, you can see this was a picture taken at Tenholt Research Station. I can't really explain where that is. And just comparing no inoculation versus different inoculation and micronutrient treatments. So we just finished a report on that, and hopefully we'll be publishing some papers on that. An area that we've been working on and recently finished a project and have a PhD student who works partly in this area, Brooke, and it's the area of biorefining, and it was specifically focused on organic production systems because high protein feeds are generally quite expensive in organic production systems. So we wanted to see whether we could produce biorefined products from forage legumes. And this involved both using a screw press, like on the right, to form juice and pulp that could be used for different types of animals and different purposes. And then we also used a leaf stripping machine, which separates the leaves from the stems. So to give you an idea what that looks like before and after harvest, it does a pretty good job, and it increases the protein content a little bit. And again, we're in the process of publishing some of that work. So biorefining is something that we've worked on a little bit. We're involved in some grazing studies, particularly with the animal science unit, which is also part of our department. So I didn't mention that the current structure of our department, we're part of 2 different faculties. And we have an animal science group and a crop science group. And the idea is to collaborate across plant-animal research topics. And grazing is an example of where we do have, well, one good current project where we're collaborating. We are not plant breeders, but we do collaborate with plant breeders on projects, both with people from Lantmännen and also people in Alnarp. And we're involved in 3 different grogrund projects, one to do with Timothy, one to do with red clover. They're both genomic selection projects, and the last is to do with red clover seed production. So increasing seed yields of red clover grown for seed production. And that's quite a new project. And we also have a new project this year, which is looking at the winter survival of different oilseed germplasm in northern Sweden. So at the moment, we can grow spring turnip grape or ribs, but it's not grown a lot. But we have a project to look at some new material of oilseed rape, rups, and then turnip rape, and lapidum, which is a new crop which has been, it's come out of the plant breeding department in Umeå. So yes, so we're involved in plant breeding projects from the agronomic side. And this is just a shot of an aerial photo of one of the plant breeding experiments to do with red clover at Ås near Östersund in Jämtland. So that's the red clover genetic selection project. In terms of nutrient management, over the years there's been a series of projects that our department has been involved in. Cecilia Palmborg is involved in the botany and nutrient recycling project, which involves mainly demonstration experiments with different digestate products. And so this is an area that CC has been working in for quite a long time. And I'm not going to go through lots of the details here. And CC has also been involved in a number of biomass production projects, which also do involve some biorefining as well. And this project, Grass Refine, which was financed by the Swedish Energy Board, is a collaboration, I believe, with BTC, which is, can't think of the acronym, but they're involved in biorefining products. And CC's had some field experiments with tall fescue and reed canary grass, which she's worked with for a number of years. We have an interest in cropping systems, and one area that I worked in quite a bit before I came to Sweden, and we've worked in this area somewhat over the years, but it's taken a little bit of a dip at the moment in terms of current projects, but it's still an area that we want to work on more, is modelling agricultural systems. So we've worked on modelling barley and oats and maize, even though we don't grow maize in northern Sweden, and also forages. So this is an area that we do have an interest in developing further projects. And we work with, primarily with the APSIM model, but with a range of different agro-ecosystem models. Also, in terms of cropping systems, we have some long-term experiments in northern Sweden. This is the R871 long-term experiment. We had a master's student, well, actually, we've had two master's students over the last few years study this experiment, which looks at the effect of the number of years of lay in a cropping system and what effect that has on the long-term sustainability in terms of the production and the soil. And there's some really interesting outputs from this long-term experiment. But we have another long-term experiment, the R874, and I won't go into the details, but that's something which is severely underused in terms of the data. We have, more than 60 years of data and very, very few publications. So that's an area that we need to strengthen to get more outputs out of our long-term experiments. So there's a good source of data there for students. An area I could say close to my heart is new forage legumes species and cultivars. This is sort of my original background when I was in my undergraduate years and one of my big interests. And some of the plants, we've tested all sorts of plants in northern Sweden, and sometimes we've just sown 30 plants planted out individually because we only have a tiny little bit of seed. And so we tested lots of different species and genotypes to see what might be an interesting plant to grow in northern Sweden. And one of the plants that has come out of this is yellow lucerne. So most of you would know blue lucerne, which is Medicago sativa. And then there's Medicago sativa subspecies falcata, which has been used to breed winter hardiness into blue lucerne. So the blue lucerne that we have in Sweden would have been crossed with yellow lucerne to develop more winter tolerant characteristics. But we can also grow yellow lucerne, and there are some existing cultivars developed in other countries. So when we saw that it was an interesting plant to look at, we developed an experiment to test different lucerne genotypes. And I should also mention that there's also what's called melon lucerne, or in English, variegated lucerne. And this sort of has the characteristics of both blue lucerne and yellow lucerne. But the reality is there's no good way to tell, to characterize, to say that's blue lucerne, that's yellow lucerne. Like, it's based on the color of the flowers, but there's a lot of other characteristics as well. So we have some field experiments at 4 different locations in northern Sweden, looking at different yellow lucerne genotypes, or different lucerne genotypes, because this includes blue lucerne and yellow lucerne and different hybrids. And here's some early results from Ås, from one of the sites. And this is the red clover control, so Umeå, which is a fairly new red clover cultivar, and producing sort of typical yields. And we've got a bunch of lucerne cultivars which were better. And I don't think it was just to do with the red clover wasn't performing well. It did perform quite well. And another thing that I want to mention here is that we were just planning to have 2 harvests because we weren't sure how the yellow lucerne would go. But in the autumn, we noticed that some of the lucerne cultivars were very, very autumn vigorous. So they were producing a lot of biomass in the autumn. And so we decided to cut it because there was a couple of tons of dry matter there, and this is what a farmer would do. They would cut that. And so these are the more blue lucerne types, when you can see we got a 3rd harvest, but we'll be interested to see this spring whether those ones which were cut late will survive the winter very well, because that can affect the winter survival. But this Carlu yellow lucerne, which is a true yellow lucerne, from 2 harvests, it still didn't have a significantly lower yield than the ones harvested 3 times. So we're getting some interesting results, and it's too sort of early to say that this will be an important new legume, but it might be. So it's something which is interesting to us. We're also interested in native legumes and improving the biodiversity of leys by adding native legumes. This is my idea of what a ley might have looked like 100 years ago, with lots of biodiversity, lots of grass species, wildflowers. As leys have been developed with higher yields and resowing, we've narrowed it down to usually a couple of grass species and 1 or 2 legume species, and often just 1 legume species. And that sort of system doesn't provide as much biodiversity or opportunity for pollinating insects that an old-fashioned biodiverse ley would provide. So we wanted to look at existing native legume species and see if we could reintroduce them into lays. So this project evaluating the potential of native forage legumes in mixed lays in northern Sweden. Yeah, I guess I've already said this, but we often in a lay, red clover will go well for the first 2 years and then not produce much anymore. So often different diseases will reduce the red clover concentration. And potentially we can introduce small amounts of native legumes. We're not trying to take over red clover or replace red clover. We're trying to add to the mix which is already there. And maybe we'll have some legumes that in longer term lays, that instead of lasting for 3 years, might last for 5 years or something like that, then we'll still have some clover or legume in those plots, providing maybe a bit of nitrogen fixation, some biodiversity and some pollination resources. And Brooke, who is a PhD student, has been working on this area and using existing botanical resources, the herbarium, floras and databases of where different species have been found, and using these resources to narrow down potential perennial legumes that we could use. And again, we won't go into all the details, but these were 7 species that were identified and then they were collected. So we went to the locations where these were growing in the wild, collected seed, and we're in a process of analysing them for their nutritive value and their potential anti-nutritional compounds, because we don't want to add things to the lay mix which could be negative to animal production. And some of these, I will admit, have the potential to be, particularly from the astragalus and the lathyrus genii. Is that the plural word for genuses? Yes, so we also have all of these, yes, I believe, all of these species are part of 2 different field experiments, which we've established at Tammsalen, which was sown last year. So this year, we'll be looking at the production of these native species in a lay. And I think it's really interesting because, I mean, it's really the first time that these species have been sown in agriculture. Another area that we work in is using sensors for farm decision support. And I'll give you some examples of some projects. We've been working with field spectrometers, particularly the ones like the Yara-N sensor. So this is, the Yara-N sensor is a commercial device which sits on the top of a tractor and was initially designed to provide precision support on nitrogen use in cereals. And what we've got there that Zhenjiang has is a handheld Yara-N sensor, and we've been able to link that to different forage quality characteristics. So like the digestibility and the fiber content. So this still needs further work, but it shows some promising results. We also had a project where, and we still want to develop this further, where we're using image analysis from simple cameras. So this was all done with an iPhone camera, and then using like artificial intelligence to classify different, or identify and classify different types of plants, to be able to estimate the botanical composition of mixed lays. And this is really important because the botanical composition in lays affects the recommended fertilizer rates. Because if you have high levels of clover, then you reduce your fertilizer rates. But the problem is there aren't really good methods for farmers to actually estimate the fertilizer rates. And I just want to show you a little video that we did about this project. But the idea was that using this image analysis technology, we could use a cheap consumer grade drone that weighs under 250 grams, so you don't need a license for the drone, and use that to scout in different areas of the field, take photos and learn some information about the field. So let me just share this video. There is supposed to be some music, but you don't really need to hear it. Yes, so I definitely think that that video is better with the music. But so the main part that I wanted to show there was the clover estimation in spring, and also the ground cover estimation in spring is really easy to do with drones. It's very, very simple. The clover content is a little bit harder, and we need more testing of that because we can't accurately control the height of the drone, and we need to get quite close to the ground. So that's something that we need to solve. We also have a project that started up recently with Umeå Technology University, where we're putting some of these sensors on an autonomous ground vehicle or a robot, and that's in the process of being assembled at the moment. And so we're using a cheap spectrometer, like the RN sensor that I mentioned before, to tell us about forage quality. And then we're using various types of cameras, including an RGB depth camera, which can develop sort of like three-dimensional photos to tell us something about the quality and the amount of biomass. So the idea is that this robot can go out by itself and scan fields. We have a new project with barley, because we always have a number of fields of barley at Tammsalen, and one of the things that we were talking about last year is these patterns that we see in the yield maps. What's causing these patterns? Because they're reducing the overall yield in our crops, and we could likely see the same things in farmers' fields. So we have a project where we want to understand the yield variability using various remote sensing methods, and then link the remote sensing data to the yield map data and do sampling of different types to determine what is causing the variation in yield. So, for example, here we've got the yield map on the left. So this comes from the harvester. And then we have an NDVI map from satellite, which is in sometime in July. Yeah, so the 25th of July. So we want to see whether we can sense where the low and high yield areas will be early in the growth of the crop and determine what is the reason. So is it due to, you see some really interesting patterns on the yield map. So what's causing those? Is it due to bad drainage? Is it due to fertilizer being put out in an inefficient manner? Is it due to compaction around the edges of the fields from tractors driving? So we want to do sampling in different yield zones so that we can understand the limitation. So this is quite a practical project. We've also had a project called Valsat, where we're using satellite data to estimate the production and quality of maize. And this is, we've collected a few years of data for this project, and we're in the process of writing that up. But this is something that we would like to develop into a tool usable by farmers, similar to the CropSat tool, which has been developed by researchers at SLU. And as a reminder, all of these remote sensing projects are made possible by people doing work in the field because we have to generate good data sets, we have to measure things in the field. And so for this project, well, we try to combine, when we do the sampling, we try to combine multiple projects in one. So I think we're sampling for 3 different projects here with different types of instruments: drone, satellite, and handheld spectrometer. So it's not all just playing around with fancy toys. When it comes to the different types of technologies, there are differences in resolution. So satellite data is often at a coarser resolution. So here's the same area sensed with a satellite versus sensed with a drone. So with the satellite, we can get, well, with the Sentinel-2 satellite, we can get 10-meter resolution, at least some of the bands. And that's really useful at a field level, but no good for assessing experimental plots. Whereas drones, it's if you have very many hectares of land, it's very difficult to assess those all with a drone, but that's where a satellite can be useful. So with experimental plots, we might, depending on the height and the resolution of the camera, we can get very good accuracy and we can see very closely what's happening. So that moves me on to the last area that I want to talk about, which is using remote sensing methods to assess agricultural field experiments. So using drones for assessment of field experiments. Often when we're flying drones, we create ortho mosaics, which have been spatially corrected and color corrected so that we can compare distances and colors across the whole photo. And so with a drone, we'll fly, and the drone will be it'll fly a predetermined path, and it'll take photos in certain places and at spaced distances apart with a specified overlap of photos. And then software is used to create the orthomosaic, which we can then use to assess the field experiment. So that's sort of the basic idea. One application of this is we've been working with Lantmännen on assessing survival of red clover plants in the spring. So it's quite easy in the spring to separate the green plants from the brown background material. So on the left we have the photos of the plots, and then in the middle we have the green cover, and then it's quite easy on a plot basis to say X% of this plot is covered by clover, assuming that we don't have any weeds. And then another method that we were working on was to put lines through the different rows of clover plants in the field and say what percentage of the lines were covered. Because what they're looking for in assessing these plots when they're done by eye, because that's the way they assess these plots at the moment. Somebody walks around and looks at every plot and looks for missing plants and says 90% or something like that. So they're looking for gaps in the lines. So we can potentially find the rows and put lines through and then say what percentage is missing. So that's an example of a project. In addition to visible light, we can also use multi-spectral sensors so that we can get more information about the quality of the plants or the status of the plants. And another thing that we can potentially do is to develop 3D models. So if we take photos from different perspectives, then this can be combined to create a model which can be sort of rotated in 3 dimensions, and we can, if we know where the ground is, we can, in a sense, estimate the volume of different plots. So, in this photo here, that's a 3-dimensional orthomosaic of a lot of different photos, and you can see that this field experiment hasn't been done super well in that at the edges of these plots there are areas which haven't been sown, and so maybe this is before the ends of the plots were cut off before harvest, so these haven't been harvested yet. So you can see the differences in the plots, and one of the things that we're interested in doing is using drones not just to compare the differences between plots, but to compare the differences within plots, to say how variable is every plot. Does it have an even coverage of biomass, or do we have variability in biomass? So giving plots a rating based on their variability, and sometimes that could help us to understand why we get these error terms in our experiments, because and hopefully explain a little bit of this unexplained error. So that's my presentation, and thanks to all the staff at NGV who have contributed to the research behind this presentation, including not just the crop science people, but all of the other groups in our department.